skeletal muscle. And so we'll start off by kind of introducing the anatomy of skeletal muscle. So I know you've looked at a lot of muscles, especially in the anatomy labs. You've seen, the tendons, you've seen the muscle bodies themselves, and you kind of have an idea that muscle shortens and contracts. But let's look at the actual anatomy of what muscle looks like under the microscope, so at a histological level. So we're speaking here about skeletal muscle. So understand, usually when we talk about muscle, we usually think about skeletal muscle. There are other types of muscle, but skeletal muscle is what we're going to start off and focus on first. So it's usually connected by 2 or more bones, by tendons. So the tendons are the non-contractile part of the muscle. So they basically insert onto bones. And if you look at a muscle body, we can see that it's covered by this connective tissue sheath. That's called the epimysium. And the epimysium is what kind of separates one muscle from another. That's how we know that the biceps is a specific muscle body. And it kind of also helps muscles to slide over each other and the nerves to also slide over those muscles, nerves and vessels in the area. If we took that muscle body and cut open into the epimysium, we would see a bunch of muscle fascicles, hundreds to thousands, depending on the actual size of that muscle. Those are covered individually by the perimysium, and those are called muscle fascicles. Okay, so those are muscle fascicles. Those bundles of muscle cells that group themselves into these fascicles and are covered by the perimysium. Now, if we went even further into the perimysium and into a muscle fascicle, we'd find a single muscle fiber, which is synonymous with a single muscle cell, although it's kind of an elongated version of a cell. And this is covered in the endomysium, another connective tissue sheath that separates a single muscle fiber from its neighbor, okay? So the endomysium covering a muscle fiber or cell, perimysium covering muscle fascicles, and then the epimysium covering the entire muscle body. Now let's talk about some specific nomenclature for the muscles. First of all, muscles are multinucleated, and this kind of sets them different from some of the other types of muscles that we'll talk about later on, like smooth muscle. Then we've got the sarcolemma. The sarcolemma is the unique name of the plasma membrane specific to skeletal muscle. Okay, the sarcoplasm likewise is the unique name of the cytoplasm or the cytosol that's unique to skeletal muscle fibers as well. And you'll hear me use the word muscle fiber and muscle cell interchangeably. They're the exact same thing. Okay, the muscle fiber is the muscle cell. All right, let's look at this here a bit closer. So we've got a muscle. Here's its tendon. Here's the origin. So this is where the muscle, this is the stable part of the muscle. And then down to the distal part, that's going to be the insertion. That's going to be the mobile or the movable part of the muscle. The movable part, or the movable attachment, I should say. If we look closer here, we can see that fascicle. So here's that bundle of fibers wrapped in the perimysium that creates a fascicle. And if we look even closer within that, we can see a muscle fiber. So this is one muscle cell, so to speak. And as we can appreciate, there are many nuclei within this muscle cell. Now, if we look even closer here, getting into the subcellular level, so beyond the level of the muscle fiber, right down into the organelles that are inside that cell, we'll see that inside a muscle fiber, there aren't many other organelles. They're just these elongated structures that are kind of jam-packed inside the muscle fiber. These are called myofibrils, okay? So again, just for reference, here is the muscle cell. And I know a lot of students have a hard time with this because it's not like a circular cell the way we would typically draw a cell. This is a cell. Inside of it, we've got all of these, myofibrils packed end to end. And so there really isn't room for many other organelles except the mitochondria. Now, we know that energy is super important, ATP specifically, for muscle contraction, for muscle movement. And so there are a lot of mitochondria that provide the energy to allow for muscle contraction. So we've got many, many myofibrils jam-packed in this cell, along with many mitochondria. And if you look at the periphery of the cell, underneath the sarcolemma or the plasma membrane here, we also see a couple other structures that are really important for muscle contraction. So there are transverse tubules. So these green structures, they're called transverse tubules, or T-tubules for short. And they're really important as it pertains to the conduction of the action potential, or the end plate potential as we've described it, okay? And so as we can see from a surface view, they're kind of wrapped around the surface of the cell or the fiber. But if we took a horizontal or a cross-sectional view, they actually run pretty deep and right through the entirety of the muscle fiber itself. Okay, so there are these deep invaginations, and that's why they're so important for the conduction of the action potential, is that they help that end plate potential go from the motor end plate where the neuron is innervating, the motor neuron, and go all the way deep into the muscle so that the action potential can spread throughout the muscle fiber really, really quickly. Okay. Now, adjacent to the T-tubules are these sacs. They're called terminal cisternae or lateral sacs. Really important for storing calcium. Calcium is going to be vital for muscle contraction, really, really important calcium. And so it's going to be stored in these lateral sacs that are adjacent to the T-tubule in the center. And together we can think about these three structures as a triad. So a triad is consisting of two lateral sacs, which are storing calcium, and the central T-tubule where the action potential or the end plate potential is being propagated throughout the muscle. Okay. Now, if we look even closer at a myofibril, right? So kind of keep up here in terms of the level of that we're looking at, right? So we talked about the muscle belly, we got the muscle fascicle, we got the muscle fiber. Now we're looking even, at a more microscopic level here at the myofibrils. And if you look at a single myofibril, these are the units that give muscle its striated appearance. They're only seen in cardiac and skeletal muscle, and they help to give muscle its striated or striped or, kind of tissue-like elongated appearance. And if we look at a myofibril, we're going to see many repeating units of these thick and thin filaments. The thick filaments are myosin, these filamentous proteins. The thin filaments are actin, which are also filamentous proteins, okay? And so the orderly arrangement or the stacking, right, the interweaving of actin, myosin, is what creates these sarcomeres. And then we've got many, many sarcomeres stacked against each other, creating this myofibril. And this is a structure that makes muscle appear the way it does, kind of striped or striated. All right, so let's speak about viewing muscle under the microscope. So before we actually look at the light and dark staining bands of a histological view of muscle, let's think about what this actually is. So first of all, no one has ever actually visualized muscle contracting under the microscope. It's just not possible. When we're looking at muscle under the microscope, it's dead muscle, okay? And so based upon what we see some dark staining regions, right? This is a sarcomere, by the way, so really, really microscopic here. We see some dark staining regions. We see some light staining regions, and we see some kind of intermediate staining regions. And so earlier physiologists, they saw this in contracted muscle, they saw this in relaxed muscle, and then they kind of theorized what had to happen to these regions in order for the muscle to go from its relaxed state to the contracted state. So basically comparing those two histologic views. And what they came up with is called a sliding filament model, right? That's why it's a model, because no one has ever actually appreciated it under the microscope. And so what they saw were that these staining regions represented different amounts of protein, so different areas of the sarcomere where there was a presence or absence of those thick and thin filaments. And so if you look at a sarcomere, there's the A band of the sarcomere. This is the area of a dark overlap between the thick and thin filaments. Okay, so here we can see the thin filaments are in orange in the center, the thick filaments are in orange in the center here. The thin filaments are in blue, and these are going to be the outer borders of a sarcomere. So one sarcomere is from one Z line to another Z line. Okay, so from blue to blue is one sarcomere. And so at the center of the sarcomere, we see these orange protein filaments. These are called the thick filaments. These are made of myosin. And because there is an area of overlap between the thick and thin filaments, in other words, the blue structures here and the red or orange structures here, we call that area the A band. And if we look at the histologic view here, it's not as light as the Z line, but it's not as dark as the center of the Z line either. And it's just this area where we have overlap. So it's kind of in the middle in terms of the staining pattern here. If we look at the center, the very center of the sarcomere, this is where we have thick filaments, so there's no overlap here. So just thick filaments form the center of the sarcomere here. That's the H zone. And at the very middle, there's one line that goes down, that's going to be the M line. So that is where the thick filaments are attached to each other. So they're forming this line, attaching to each other along the center of the sarcomere. And then so that's the H zone, and at the center of the H zone is the M line, okay? So these are the central staining patterns of the sarcomere histologically. The A band, which represents the area of overlap, the M line, which is just the attachment points where myosin filaments are attached to each other, and then the H zone, which is just the outer periphery of the myosin filaments, which is not overlapping to the actin just yet. Let's look at some of the outer regions here. So we've got the I band, and the I band is just actin filaments, okay? This is kind of lighter staining because these filaments are smaller. We'll look at the actual structure of an actin filament in a bit, but they're smaller structures, they're thinner proteins, hence the word thin filament. And so they're going to provide a lighter staining region around the periphery of the sarcomere. These are called the I bands, okay? And again, there's no overlap here between thick and thin filament. This is just thin filament. The Z line, however, is the link between each of those thin filaments that form this line down the center. So the Z line is kind of synonymous with the M line being the point of attachment between the thick filaments along the center of the sarcomere, the Z line being the points of attachment between the thin filaments along the periphery of a sarcomere. And so based upon those appreciations, those structures, we can consider one sarcomere being the functional unit in between one Z line and another Z line. Okay. And we can also think about a cross-section of a myofibril here. So if you look at this cross-section of a myofibril, the A band is where there's thick and thin filament overlap, right? And then the very small dots or the small cross or cut surfaces are representing just the thin filaments, and the larger specks are representing the thick filaments. So basically, in order to get this cross section, I would have to take it somewhere across the A band. This is the only place that I'm going to see both the thick filament and the thin filament. And if I look at that cross section more closely, I can appreciate that the thick filament is a larger cut surface, a larger cross surface there, and the thin filament is a much thinner protein, so we can see a thinner or smaller cross section. Now, let's talk about the structures themselves, the filaments themselves. So we've got the thin filament, which is really actin. It's a contractile protein, and it's got these binding sites for myosin. Okay, so here's an actin monomer, and it's called G-actin because it's a globular protein, which is really a rounded or round-shaped protein. And then these globular proteins aggregate themselves to form this actin strand. This is called F-actin because it's now a fibrous or elongated protein, okay? And when we have two F-actin proteins, we can then intertwine them together or kind of wrap them around each other. And this is what creates our actual actin strand, okay? So it's really these G-actin monomers, joining together like pearls on a string to form this F-actin monomer, and then two F-actin monomers intertwine themselves, I can never say that word, intertwine themselves to form this double helical strand, which is our actin protein, our actin strand, our thin filament. Now, if we look at some of the other proteins that are involved here, so there are other regulatory proteins. There's tropomyosin, which is overlapping the binding sites on these actin monomers for myosin. So it's kind of slow down here. So basically, we've got the binding sites for myosin to bind. Remember, myosin is the other thick filament, the other filamentous protein that we talked about. It's at the center of the sarcomere. And so the binding sites are usually covered up. That is to say that myosin doesn't always have access to bind to actin. That's how we regulate this whole process, okay? So tropomyosin is covering up those binding sites. It's this thread-like protein that's wrapped around the binding sites. Remember, these black dots are the binding sites. The second regulatory protein here is troponin, and troponin is really a complex of 3 attaching proteins that have 3 different binding sites. So we can see this pink structure here. It's got 3 bubbles or 3 different protein structures attached. One is going to be an attachment point to actin. So that's the first one at the bottom here. One is going to be an attachment site for tropomyosin. Remember, tropomyosin is the thread-like protein that's wrapped around the actin strand. And then the third site is going to be where calcium binds and can actually stimulate or regulate or start the muscle contraction, okay? So it's really important for calcium to be present because when calcium binds to the troponin complex, it's going to allow the entire complex to drag the tropomyosin threads off of those binding sites and pretty much expose those binding sites so that myosin can bind and initiate muscle contraction. And so that's why we call tropomyosin and troponin the regulatory proteins. These are the proteins that are going to turn this process on and also turn this process off by the binding of calcium. So calcium is what's going to drive this whole process. Really, really important to understand that here. All right. So that's the structure of actin, a little bit more complicated. Let's look at the structure of myosin, or the thick filament. So myosin really is located along the center of the sarcomere. So we pointed it out. It's connected, they're connected right down the middle by the M line, and they're really forming the H zone of the sarcomere. They're going to extend all the way out to the I band. They're not a part of the I band, but that's where they end. And when we look at the actual structure of myosin, it also has a binding site to actin. So these 2 binding sites will go and bind to this site here, right? So this site on the actin. And so we've also got another binding site here for ATP. This is called the ATPase site. And so the myosin head is really the movable or the part of the sarcomere that's going to energize the muscle contraction. So ATP will bind onto the ATPase site of the myosin heads. And that will then allow the actin sites on myosin to bind to the myosin sites on actin. Kind of weird here, right? So myosin has the actin binding site, actin has the myosin binding site. And when ATP binds to this myosin ATPase site, those 2 connections can happen, provided that there's calcium present to expose those binding sites on actin. Okay, and we'll talk more about that a little bit later on, but just kind of making the connections of how these binding sites are interacting with each other. And when we look at the actual structure of the myosin or the thick filament, it's really, again, a wrapping around or an intertwining of 2 of these proteins that wrap around each other, almost like 2 golf clubs. And they extend outward from the center of the sarcomere towards the area where actin is located. Okay. Another important protein found in the sarcomere is called titin. Titin is an elastic or stretchy protein, and it's found at the tips of the thick filaments extending toward the Z line, which are the borders of the sarcomere itself, okay? So the purpose of titin is really to anchor and provide support as the sarcomere shortens and contracts, and it's going to help the rebound of the sarcomere when the shortening and lengthening of the sarcomere are occurring. So it's really providing the structural support, this stretchy nature or this elasticity to the sarcomere to really facilitate that contraction of the sarcomere. Okay, now before we actually illustrate the sarcomere here, I want to just kind of talk about what happens when muscle contraction is going on, what's going on in the sarcomere itself. So someone is asking, is acetylcholine the neurotransmitter of myosin? So yes, acetylcholine is the neurotransmitter of muscle, of skeletal muscle at the neuromuscular junction, right? So not necessarily myosin, but at the neuromuscular junction at the motor end plate. And it's going to bind to only, remember, there are only nicotinic receptors on skeletal muscle. And that is what's going to bring about the sequence of events that we're going to talk about now. Okay. So before we illustrate this, I want to point out that when the muscle contracts, the contraction is really happening all the way down at this microscopic level of the sarcomere. So the sarcomere is what's shortening to bring about the entire muscle contraction, okay? And when we think about all types of muscle movement, so any kind of muscle movement, muscles of the face, muscles of the body, the limbs, when they move, what we're really seeing is that these thick and thin filaments are sliding past each other. That allows the shortening of the sarcomere. When we have the shortening of the sarcomere, the entire myofibril, remember the myofibril is nothing but the repeating units of sarcomeres. So the shortening of one sarcomere brings about the shortening of the entire myofibril. That brings about the shortening of the entire muscle fascicle, which shortens the entire muscle body, right? So this is really a contraction which is happening all the way down at the histological level of the sarcomere. And that's why we can consider the sarcomere as the functional unit of muscle contraction.